That's my cue. Hi, you guys. This is Pam Winfrey. I'm the host for the ASU Laser, Leonardo Laser, which, as we've just been talking about, stands for Leonardo Art and Science Evening Rendezvous, which, as we know, time is relative. So for some of us, it's the evening, and for some of us, we're just barely waking up. So I appreciate whatever time zone you guys are in. Today's topic is facility of mind, how thinking in new ways can inspire new questions. And basically, that's just this idea that when you think, when you work, when you play with people who don't think, work, and play the way that you do, interesting things come up. And in this case, we're going to use a case of that includes Boredom Research, that is Vicky, Isley, and Paul Smith, who are from Britain, and they came in residency and they collaborated with the Arizona Cancer and Evolution Center, which is in Biodesign. Is run by Dr. Carlo Maley and Dr. Athena Actipis. And I immediately, when I met Paul and Vicky, I started learning new things because they brought me Bovril. And so I found a whole new way of thinking about food. This is food, it's in a bottle, and I have yet to try it. I must admit, because I'm a little bit scared of it, but I understand that it is a new way of thinking in a bottle. So thank you, Paul and Vicky, for that. So anyway, so when Paul and Vicky came to Arizona State University in Biodesign and ACE, they met this really wonderful biologist. His name was Angelo Fortunato, and he's working with model organisms such as sponges and Placozoa. And there was something about the combination of these minds. They just met, and all of a sudden it was like this wonderful thing happened. And I always think about this art science conversation as being at the ocean. You know, you have the land and you have the ocean, and everybody's right there because that's where all the dynamism is, right? That's where it's exciting. That's where the conversation starts really rocking and rolling. So I feel like this residency is a primary example of that. So then we decided, well, we have to show this someplace. So we went to the ASU Art Museum, and Heather Lineberry, who is the curator, senior curator, and Brittney both said, yes, we're in it, both feet. And I was like, this is great. We can do this one piece. And they went, no, we'd like to do a whole bunch of their pieces. So in the end, we're doing the entire bottom floor of the ASU Art Museum. And today we're going to hear from not only Boredom Research, but also Angelo and Brittney, which means we get to hear, as we were talking earlier before this started, Vicky was saying that it's very unusual when you can actually hear from the artists, the scientists, and the curators. So that's really exciting. Each panelist has 10 minutes. And then what we'll do, which is even really the critical part of this, is the half an hour after that where we can really talk together. Because what we're trying to do is create a community of thinkers and doers and people that can potentially collaborate together. So that's really the purpose of these monthly meetings, is to try to get people to have synergy and collaborative opportunities. So we're going to start with Vicky and Paul, also known as Boredom Research. They've been collaborating with world-class science institutions since 2014. And I have to say, I feel as though they are world-class artists collaborating with world-class scientists to create world-class work. So with that wonderful introduction, I think we can turn it over to Paul and Vicky.
Well, no pressure then. Hopefully we'll give a world-class talk as well. Okay, we're just going to have a go at sharing our screen. Okay, that should be good. Okay, so you should all be able to see in Search of Kimazos and a nice image of Kimazos. So yeah, thanks for that, Pam. And like, I did notice the Bovril didn't look like it was open. So we'll send you some recipes later, but we've only got 10 minutes now, so we'll get to it. So myself and Paul have been collaborating with scientists, as Pam explained, worldwide, but mainly to enhance engagement with our world. And we feel like balancing the insights of science with the sensibility of the non-expert is important in our work. and also reducing the dangerous void that can separate these spaces as well. So we're very much interested in interdisciplinary research, but how we can look at it from the standpoint of art, science, and society as well. Hello. Would like to move on.
Right, there you go.
Okay, so over the last 20 years, Boredom Research's artwork has explored topics such as the fragility of natural systems. So in this artwork that you can see here, robots in distress.
And we've also kind of very much kind of interested in the mechanics of the natural world and thinking about the world in quite a mechanical way. And here with a project that we did, we created a loom that generates a diversity of species. So it's thinking about those mechanics as, you know, on one hand, a product of diversity, but also in the case of the textile industry, something that kind of impacted on that diversity. And another project working with an epidemiologist that was looking at disease transmission, but from a landscape perspective. So, you know, very much kind of thinking about zoonosis and kind of zoonosis in a landscape that's really kind of engineered by the palm oil industry.
Okay, so we've always, in all our work since day one, we've always been fascinated by natural systems. And so when Pam invited us to work on this collaborative project with the Biodesign Institute, we very quickly became aware that their philosophy was all about being inspired by nature, by learning from nature. So we just wanted to kind of jump on this opportunity because it was an ideal opportunity for us to work, you know, thinking about biomedical research, but also like natural systems as well. So the collaboration started in, as Pam explained, in 2018, where we started collaborating with the Arizona and Cancer Evolution Center. And we were really excited to be working with the cancer researchers there who were learning from natural systems to enable them to do not only better cancer prevention, but also better cancer management as well. And of particular interest to both Paul and myself was how at that center, they were looking at systems from not only a biological perspective, but also an ecological perspective as well. And that was something that we really picked up on, you know, quite early on, especially when we were talking to Angelo that we ended up collaborating with, because he was developing new model organisms to study cancer.
So Angelo kind of, he took us into his lab and he kind of introduced us to the Placozoa, this kind of, this tiny magical organism, which he put under the microscope. And one of the things that really kind of moved us, we'd been immersed in this kind of, in this conceptual space that was thinking about cancer, something that happens at the level of the cell. And here we could see a being that we could see it's the whole of its body and its individual cells at the same time, so that we could see both we could see it both at the level of the mechanics of the disease, but also at the level where we could consider it as a single entity.
So it was this organism, the Placozoa, that inspired the Chemazoa. This is where the Chemazoa was born, from that moment when we actually saw the Placozoa. So the Chemazoa in our artwork that has been commissioned by ACE is called In Search of Kimazos. So that's the title of the piece. And it represents a fictional organism that experiences the same disease processes that touches all of us. I think it's probably fair to say that we've all been touched with someone that we know, someone that's got cancer, or we've, you know, experienced it in some way. So the artwork responds to new therapeutic approaches centered on managing rather than curing cancer. So I'm going to have a go now at sharing the video teaser of the work. So it's very short, it's just over a minute. And in the teaser, it's got elements of the documentary and also the multi-screen moving image installation as well that will be screening both works, both the documentary and the multi-screen piece at ASU Art Museum. And the exhibition launches on the 5th of December, so quite soon. So they're in the process of installing this piece.
Something had changed. There are a great many uncanny sounds below the water's surface, and now, among them, a new chorus.
So I think one vision of the future is that everybody will get cancer at some point in their lives, but you'll just live with it. And so it'll just be a normal part of a stage of life.
Angelo has managed to cultivate some amazing organisms in the lab. They're simple multicellularity. They don't have as many tissues as we do.
How has nature discovered ways of preventing cancer, and then can we translate that into doing better cancer prevention in humans?
Why these animals, they are not developing cancer, and that they have something that we don't know.
The pale song of a languid sea. A song made of a million frail voices united in a search.
So, so the time that we spent at ACE was really kind of quite. In a way, we came away really quite aware that it was quite an exceptional experience. That we was kind of exposed to what we could probably maybe think of as a really dendritic thought process. That a lot of the research that we was experiencing was being kind of fed from so many different directions, and considering the subject area from kind of very different scales of reference. I mean, in particular, Angelo was kind of based in these two labs, one kind of centered on computational modeling, and the other one approaching cancer from the perspective of psychology. And also he was kind of coming to that subject, but from, coming to working in the context of, in a biomedical context, but bringing to that an ecological understanding, so that it was such a complex research environment that it kind of really forced us to produce what is for us quite a different work, and to work in a very different way to the way that we had been working in previously.
So often we work where we just create a computer simulation, but for this work, there was so many, it was so rich and diverse, the different layers, like Paul was describing, of what was being inputted into the work, that we wanted to incorporate the computer simulation, but also filmed environments to show the importance of these ecological natural environments as well, for not only scientific research, but for human health as well. So in Search of Kimazos combines as a 3-screen, multi-screen moving image installation. So there's 3 films that play simultaneously. And there's elements in those films that are the computer simulation, but also filmed environments. And there's a completely new process that we use. So we use a voiceover artist who has a godlike narration over this piece of work. That by combining all these elements, it kind of enabled us to combine the breadth of the research that was going on at the Arizona Cancer and Evolution Center. So with the dynamics of cancer programmed into their cells, the chemozoa are designed to survive in toxic environments. And the toxic environment that we're depicting is an analog for chemotherapy. So the artificial physiology of the chemozoa does not differentiate between healthy and unhealthy cells. And therefore there is no clear distinction that can be made between cancer and body. And as such, the chemozoa escape the existential crisis of experiencing an internal conflict between healthy and unhealthy self and other, which are all kind of characteristics of cancer.
So, I mean, one of the things in terms of, you know, really kind of the impact it had in our thinking is that we were very, you know, we went into that situation very kind of, you know, engaging with the idea of science inspired by nature, the idea that we can look to the natural world to solve some of the problems that humans are facing. And then we kind of started to think of that very much as how can we understand that more as a conversation. So, you know, for example, there's lots of things in the film where we have kind of literal reflections, where here we can see Angelo both as a scientist, but also he's kind of reflected, so he kind of appears below the water or immersed in that environment. But, you know, thinking about this conversation, it's like if we really look into nature to inform us and to kind of give us tools with which we can target challenges, and especially the challenges of human health, then how should we reply? How do we reply? How do we show that we're listening? So this kind of really brings us to the final slide, hopefully. So for us, the missing link when we kind of think about human health is that we shouldn't just consider it in terms of the cells and the behavior of the cells inside our bodies, but that our bodies are really kind of cells within a kind of an animate and complex system, and one that we have really kind of only just started to understand. So for us, if we look to kind of benefit from, you know, what we can maybe think of as the more than human world, then we have to think of that as a balanced exchange. And that, you know, if nature can allow us to live lives unencumbered by cancer, then how should we express our thanks?
And like, I feel like the ASU Art Museum exhibition, which is titled Restless Balance, kind of not only covers some of these things that we're trying to incorporate in Search of Kimazos, but some of the fragility aspects in our work has kind of emerged in that exhibition, because there's kind of pieces from 2017 to the present day. So you kind of get a sense of that body of work emerging. But that's kind of where we're at now, restless balance.
I love those two words juxtaposed against each other, because they're very, they're antithetical, and yet somehow they mesh in a nice way. So why don't we move on to Angelo? So Dr. Angelo Fortunato is an assistant research professor, and his research focuses on cancer evolution, cancer biology, evolutionary biology, and molecular genetics. He's working on human cancer and the development of novel model organisms in cancer research. Oh, one other thing, I do want to encourage people to introduce themselves in the chat, and also that's where your questions will be coming up when we get done with the presentations. So Angelo, I'm turning it over to you.
Okay, I will share a few slides. Okay, so I would like to say a few things about the protagonists of this story, that are the Placozoa. Trichoplax adherens is the name of the species, and is an interesting animal because it's the simplest animal ever described, and they have very few type of cells, and they don't have, for example, neurons. They don't have muscle cells. And they don't have a digestive system. In fact, they are releasing enzyme on the substrate in order to digest the algae, and then they are absorbing the algae. So it's a very simple organism, and probably it has more than 500 millions of years of age. Probably is evolved between 500 millions and 800 millions of years ago. So despite the fact that is so simple, or because it's so simple, maybe we can learn something about the biology. Let's see if I can. And we can use this very simple organism to address some questions about the evolution, for example, evolution of social behavior or ecological question, but also cancer biology. And I wanted to show you a couple of examples of the application or use this model organism to learn something about biology. So these slides show the behavior of these animals when they are eating, and they are able to eat together. They have this social feeding, and we discovered recently this kind of behavior. You can see here in these pictures in the left, there is many of these animals, and you can see the age of the animals, the margin of the animals. So they are this kind of greenish brown lines, and all the animals are aggregated in this group, and they are moving in the direction of the black arrows, and they are eating the algae. In fact, you see that behind these animals, nothing or very few algae are left. So they organize this kind of behavior in order to have a social feeding. And here you have pictures of a few of these animals. For example, this is one of the Placozoa, this is another, and this is another one. So you see that they are so very close attached each other. And here there is a fast motion video that show the behavior of these animals. And if I stop here, you see these whitish lines are the border of the animals, and they are very tight together. And here there is a hole in this structure. You see one of these Placozoa that is going there and fill the hole. So it's interesting that an animal that they don't have a nervous system is able to have this kind of very complex social behavior. So it's not a surprise that social behavior can emerge without a nervous system, because we have amoeba that can do something like that, like the slime molds. But in the case of a kind of complex small animal comparing to the amoeba, it's a surprise that can do this kind of behavior. So that is an example of how a very simple organism can help us to learn something about the social biology and the evolution of social biology. If you think that these animals is a very ancient model of life, and the fact that they have a social behavior means that the social behavior is something that emerged very early in the evolution of animal life. But if we go forward and we are trying to look at something cancer related, we choose these animals because we don't have any report of cancer development in Placozoa. And there are a few other group of animals that apparently they are resistant to cancer, are the hemichordata, sponges, and the comb jellies. And so the question was, these animals are really resistant to cancer, or nobody actually was able to see that until now. So we decided to do some experiment and treating the animal with the X-ray, and to see if inducing DNA damage, we can induce the development of cancer in these animals. And the first thing that we observe is that they are very resistant to the X-ray. In fact, they can be resistant to over 200 gray of radiation, and the mouse will die with 10 gray. So they are extremely resistant. But actually, from a DNA point of view, they are fragile, because the DNA after the treatment is really high fragmented. And we observed that after a couple of months of the treatment, still the animals are really full of mutations. But despite that, they look perfectly normal. And we saw something that is interesting too in the kind of behavior of the animals. So you see that there is something here attached to the main body of the animals, that is this one. And these animals, they have a different strategy of reproduction, and usually the reproduction it happens in the lab asexually. And so the animal, they can divide in 2 parts or in more parts, and they originate in this way different other offsprings. But they have another strategy that is releasing just small pieces of the animal, is a kind of budding process, and probably is not the main strategy for reproduction, but they have this in their behavioral repertoire. But here you can see that actually this small part, there is something different, is something darker. These cells, they look different. And in fact, they release these small pieces of the animals. And you can see here in the picture B, after a few hours that we will be able to follow this individual Placozoa. And after today, you see that is reduced in size. It's only the dark cell are left. And this, at this point, this small extrusion, as we called, died. And this is something that it doesn't happen just in one case. It's something that it happens very often. We have a number of statistical evidence that is true. So basically, our interpretation is that they are able to extrude part of the body that probably contain damaged cells. And so is a tissue or organism level of defense against cancer, or anyway, is a way to remove cells that probably are damaged. And this seems to be something peculiar of this animal, but actually there are evidence that it happens something also in the human epithelium. And the human epithelium, they are using this strategy to remove damaged cells. And the fact that is not well understand this process is because it's very difficult to study in vivo this process in mammals, in human or in mouse. Let's, for example, think what happened in the intestine of a mouse that develop a tumor, and you can imagine that it happened a process like that, but it's very difficult to study. And the Placozoa, this very simple, ancient organism, can give us the opportunity to learn something about a cancer defense mechanism by using a very simple model that we can easily use in the lab. So in conclusion, the point is that we can learn something by using a very simple organism like a Placozoa or sponges or other very simple organism about very critical mechanism in biology. Some of these mechanisms are involved in diseases as cancer. And it's important to be able to do this kind of connection between something that apparently look like a zoological curiosity with something that have some application in medicine. And this connection is not always easy to do for the people, because when you think of cancer, you don't think that you can learn something about cancer studying an organism so simple and so far from humans. But actually, from molecular point of view, or from a tissue point of view, there is a lot that you can learn. And in fact, there are, for example, many genes that are involved in cancer that are present in this animal, are well conserved in evolution. And we did some analysis, and we showed that these genes actually are activated after that we induce DNA damage. And these genes that are involved in DNA repair, obviously they have a very critical role in this animal to repair the damage that we induced by the X-ray. But most of these genes are the same genes that are present in human too. So I think it's important to be able to use a very simple organism to learn something about the biology and cancer. And that was my introduction to this small animal. And if you have some curiosity, I will be happy to answer.
That's amazing work, Angelo. I just learned that flies have cancer, which I was amazed at. For some reason, you're quite right. We think of cancer as strictly a human thing or a mammal thing, but actually, if you're a multicellular animal, you often get it. So,
yeah, it's true that the fly, they have cancer, but usually fly, they don't have very long life. So it's really not very easy to study cancer in fly. In the same time, you want an organism that have a somatic reproduction of cells. Most of the, not most, but many of the simple organisms, including the insects, they have a very limited number of somatic divisions. So it's very difficult to study cancer in this organism. If you think, for example, a very important, a classic model organism like the nematode C. elegans, that they are very critical to study very important process and to learn important process in development or molecular biology, they don't have at all this somatic proliferation. So for this reason that looking at different model is important, because we need to find organisms that have a somatic division of cells. Otherwise, it's impossible to study cancer, because cancer is a disease of somatic cells. So that is critical.
Interesting. We'll move on to Brittney Krallis. She's a curator at the Arizona State University Art Museum in Tempe. We know this. At the museum, she oversees and interprets the encyclopedic collection of over 6,000 works on paper. She's involved in exhibitions, programming, education, collections care, and new acquisitions and works with numerous emerging and internationally recognized artists. Her research interests include feminist art, architecture and design, curatorial practice, and the shifting role of the Museum of the Future. So Brittney, are you there? Yes, you are. So it's your turn to talk now.
Thank you, Pam. Can everyone see my screen?
Yep.
Wonderful. So Pam asked us to imagine a Venn diagram between artists and scientists. And from my experience, there are more similarities than differences. Both are driven by a curiosity and unanswered questions as they seek to understand and interpret the world around us. As a contemporary art curator, I've had a front row seat to witness how artists and scientists make natural partners. And at ASU Art Museum, part of our mission is to be a meeting point for the exchange of new ideas. In this way, we see the museum itself as a kind of laboratory. In service of this goal, we frequently build collaborations across disciplines. And in the past, we have worked with researchers working in engineering, computer programming, biology, physics, life sciences, and meteorology, among others. And I think that it's clear that artists serve as great communicators and interpreters of scientific research, but more than that, artists can be great thought partners in navigating the unknown. So before I talk about my experience working with Vicky and Paul, I thought it would be important to back up and give you some context for how this has worked in the past, because it's sometimes difficult for people to imagine how these collaborations evolve and what they look like. So today I'll briefly touch on 3 case studies from our work at the museum. Trout Fishing in America and Other Stories, which was curated by Heather Lineberry and Ron Brolio from ASU's Global Institute of Sustainability, featured work by 2 artists, Bryndis Snabornstoder and Mark Wilson, who spent 2 years following the fieldwork of conservation biologists and their efforts to save 2 endangered species, the humpback chub and its fellow Grand Canyon resident, the California condor. The artists worked with researchers at the National Park Service and ASU's School of Life Sciences. The result was a kind of vertical slice of the Grand Canyon. Their installation of photographs, videos, and sculpture explored the complexity of human-animal interactions and their combined impact on ecologies. The resulting exhibition was at times humorous, but also surprising and moving. It included a species wall that listed every recorded species living in the Grand Canyon, color-coded into different classes of species, and you'll see human being in red here. It towered over the visitors and resembled a cliff like the Grand Canyon. And the artist also photographed the bodies of dead California condors that were killed by lead poisoning and have been frozen in labs at the Center for Biological Diversity in Tucson. Each of these photographs was accompanied by a surprisingly poignant narrative written by the scientists who tracked them for years and became very familiar with them. The artist here transformed raw data and field experience into a visual representation of a complex issue. So this is a perfect example of how we work with scientists. They really fostered a sense of environmental stewardship in those who experienced the exhibition as they explored the ripple effects of scientific conservation initiatives in Arizona. This next project is by the artist collective Interspecifics. This project was called Speculative Communications, and it was done in collaboration with an artist residency exchange program from Guadalajara. And the artists, Leslie Garcia and Paloma Lopez, worked with scientists at Biodesign to translate the bioelectrical activity of different organisms into sound. They used DIY and custom-made sets of hardware that in which they harnessed the electrical energy in bacteria to amplify its sonic qualities. In their own words, the artists called this project a concert performed by bacteria. The project culminated in a one-night performance, an immersive art installation in which the audience could experience the fluctuations in energy of the bacteria as the main source of sound. And this project illustrates the underlying order within the universe and human consciousness that appears to be intimately related to vibration. I'll show a quick clip. And I've included a quote from one of the artists in this group. Every artist has a little bit of science in them, and every scientist has a little bit of art in them. This last project is called De Comiso, which was curated by Julio Cesar Morales and includes artists Guillermo Favovich and Nicholas Goldberg, who collaborated with the Center for Meteorite Studies and the Center for Solid State Science at ASU. The research investigated the cultural impact of the 4,000-year-old Campo del Cielo meteorites in Argentina. The meteorites are protected by a legislation from the surrounding province, which prohibits their export from the region, so they're often sold on the black market. For this project, they collaborated with the ASU Art Museum in examining a meteorite that we acquired. They asked questions such as, who can claim ownership of objects that fall from the sky? And in this installation of photography, video, sound, and ephemera, the artists show how they convinced state officials to allow them to classify and document over 400 meteorites. Working with the scientists here at ASU, above are some images that were captured by an electron microscope. On the left is an S diffraction pattern. On the right is a ultra high magnification image in which you can actually observe the atoms in the material and the diffraction patterns that provide information on the direction and distances that occur within the atomic arrangement. And at ASU Center for Meteorite Studies, the team also harvested images from thin slices of the meteorite under a microscope, capturing the brilliant silicate inclusions. You can see them all here. So that brings us to Boredom Research. And finally, this year I've had the pleasure of working with Vicky and Paul as one of the curators of this exhibition, which is set to open on December 5th. And the artists have already spoken in length about Chemozoa, so I'll keep this brief, but I want to just say that working with Paul and Vicky was a unique experience for me for many reasons. I've never worked with artists who have such a deep understanding of science and pure love for science. In fact, I'm tempted to call them another species of artist. They straddle this world and can interpret the language of science so well that at times I almost forgot they weren't trained scientists. And I was asking them scientific questions as if they were the scientists producing the research. But at the same time, they also speak the language of artists. They were equally as engaged with the aesthetic and sonic and poetic qualities of the work, and really served as a bridge to communicate these complex ideas. It was also a meaningful experience because the management of this exhibition occurred at the time of an outbreak of a global pandemic. And this was at the forefront of all of our minds. So I think dealing with this concept of ecological balance on a regular basis and being able to work through these conversations with these artists was just an unforgettable experience. So I think I'll leave it at that and open it up to more discussion. I have plenty to say about this experience working with Vicky and Paul, and I'm just, it's been a great pleasure working with all of you, including Pam as well. Thank you.
Thank you so much, Brittney. I think that was a great lead in to the kind of conversation that we're interested in having now. I was thinking, I love this idea of Vicky and Paul being another species of artists. And I think that it's possible that Angelo is another species of scientist, which means maybe that's why they can collaborate so well together, because not all scientists are interested in working with artists. Not all artists are interested in working with scientists. But I feel like those three actually, they got something out of working together. And I just wondered if there was any way that you could articulate what you got out of working with each other.
It's really hard to put into words, actually. But I think you've kind of like hit it on the head. Like, I mean, it was mainly because Angelo is working not only with biological health, but he has a really strong understanding of ecology as well, and ecological systems that kind of pulled us into his research, because it was those two things combined, wasn't it?
When we think of scientists, we normally, there's a tendency to think about kind of research and the need for it to favor depth over breadth. And I think increasingly there's a kind of importance for scientists to have an equal balance between the two, to both, have that kind of that focus within a subject to really understand the intricacies of a particular area, but also have like those arms that kind of reach out and draw in information from other areas. And that was something that we really felt was very kind of present in the way that Angelo moved, even in the presentation the brief presentation that we just saw, we kind of went from kind of social feeding behavior to the mechanics of cancer fluidly, without even realizing, without even seeing the kind of transition. And I think that's something that's like not only kind of, quite exceptional, but so important, because of all the problems that all of the big kind of issues that we're kind of wrestling with now. They're actually kind of, they're all joined up, we're all part of a very kind of big interconnected system, and it's only that ability to really kind of form those connections that's going to allow us to really kind of progress in the way we think about things. And that also means kind of crossing the boundaries between the sort of weird ways that scientists work and the weird ways that artists work, and allowing kind of everyone to be part of that of this kind of evolution of thought.
Yeah, and Angelo, you're the one of the first members of the lab that actually sent me. You sent me this wonderful biological. It was this image, and the question was whether it was art or science, and it turned out to be a biological image. But I couldn't tell. It was so beautiful and aesthetic, and I think you also have an appreciation for aesthetics, which really helps in these kinds of relationships. I mean, would you agree with that, Angelo?
Yeah, definitely. I really like the visual representation, the visual art, and the photography, and I really like documentaries. So I think it was natural for me to collaborate in this project because it's something that I really like. I really like to see the high quality of the images. And I try to get good quality images from the work that we are doing to try to have some kind of nice picture to show. It's not just science, but I think the quality of the images is important too. And I think the science benefit from the quality of the representation of science, as in this case, in this documentary. So I was very happy to have this collaboration.
I mean, there is some argument that you can't, it's really hard to understand what you're doing unless you can visualize it in some way, right?
Sure.
The visualization is actually it's a pathway to discovery, both scientific and artistic, as Brittney sort of alluded to.
Yeah,
that was
and,
another thing about about
okay.
Angelo's research, because you observe the Placozoa, but also you draw their movement. So Angelo does it all by hand, don't you? Do you want to explain your process? Because it's really beautiful. Like Angelo's drawings are pieces of art in themselves, aren't they?
I'm not sure if it's art, but it was useful. So basically, it was a way to track the position of the animal in an aquarium tank. And I was interested to know how this front of animals were moving. And so I think one of the way it was just to draw in some plastic sheet the position of the animals at different time. And so in the end, I have this kind of a draw of thousands of small dots with different color according to the day of the time. And it was useful to get data. So the point was to be able to track easily that. And considering the kind of light, the aquarium tank, it was almost impossible to do something with the camera or with just a picture. So the easy solution was to draw that. And I think it was something that was done maybe 100 years ago, but still it's working. So
yeah.
I think it's really interesting, isn't it? Because like you know, there's a tendency. I feel like there's a tendency in science that really kind of to lean towards mechanical processes because it takes away the hand of the individual. That there's the idea that if you do something through a manual process, that it kind of opens it up to a kind of a weakness in a sense. But on the other hand, that it seemed that very much that kind of process of drawing the movement was really about being there and about kind of that you were then experiencing the movement. So it was a kind of a pathway for you to experience what was happening in that system. And it was the experience of that then kind of informed a way of thinking about that. And I think that's quite an unusual, or maybe an increasingly unusual way of working in a scientific context.
And it quite definitely into the sort of the realm of systems aesthetics, which is, you know, sort of got a long tradition in visual art, but this you know, this tracking of movement, or, you know, tracking of changes, as a hand drawing, it does, you know, it has its own aesthetic, and you really do enter the system in that way, and I think invite viewers and, you know, people that are trying to understand it at the same time while you're trying to understand it through your own drawing.
You know,
Yeah.
we haven't really talked about data either, because they visualize data, and I had this epiphany a couple of weeks ago. We were in the lab, and there was a student who was a statistician, and then there were a lot of cancer biologists. And the statistician, we were looking at a chart with, I think it was about cancer in across species. And it was fascinating because there were these outliers up here. And the statistician's tendency was to say, well, we'll get rid of those outliers because we really want to look at the mean. We really want to look at the average. And then one of the cancer biologists said, oh, it was Carlo, I think. He said, oh, well, we're really interested in the outliers because we can get so much more information from the outliers. So I wondered if anybody had any thoughts about data and how data kind of informs anybody's work. And I encourage, Lori, thank you for adding in there. I'd encourage anybody to talk that has something to say about that, or any other topic for that matter.
Yeah, I wanted just to add a few words about the topics that we just talked about, the fact to be able to be part of the system that you are studying by drawing or by, for example, looking at these animals under the microscope. So there is a point where you are not anymore outside the petri dishes, but you are inside the petri dishes, and you are living with these animals. And I think it is a key factor, because it's at this point that you start to understand the behavior of these animals, what happened, because you are part of this world that usually is outside of us. And I think this is a kind of mental processing of that is important in science, because in the end, if you wanted to learn really something, you have to be part of this. Otherwise, you have a kind of external view of the facts, and you want it to be inside the fact. And it's interesting, it happened to everybody. At some point I thought, okay, I have a problem, but actually I discovered that everybody have the same experience. So if you spend a couple hours looking at something under the microscope, at some point you have some kind of vision after, I don't know, when you are at dinner, for example, and you see for a moment exactly the same stuff that you were looking under the microscope. So there is this very interesting mental processing that is becoming kind of part of you, and is still floating in your brain after hours that you stop to work with that. And I think is because you enter so deeply on what you are looking that is becoming a part of your neuronal processing of stuff. And it's something that it happened to everybody. Everybody that start to work, for example, by using a microscope, spend a lot of time. At some point you see cells, maybe you drive and you see a bunch of cells, etc. So it's a kind of process that I think is interesting.
I've experienced that exact process. I'm an artist, but I ended up in these cancer research labs at a young age. And so I was an artist surrounded by so much science, but this, looking at cancer biology underneath a microscope, I was able to see the perpetuation of pattern between micro and macro. And it's a really, really profound relationship between the two. You can look within and then you see without. And so right now I'm a student at the Desma School of Design Media Arts at UCLA, and I'm studying a thesis comparing the cancer in the body to humans on the earth and exploitative capitalism specifically. And so that's what I'm investigating. And it's really kismet and pretty incredible to hear your guys' work, because it fits in exactly with the thesis that I'm working on. So thank you so much for everything you guys are doing.
Jennifer,
great. It sounds like your research has a lot of synergy. Yeah, and we totally felt that as well when we were looking at the Placozoa. It was like, and that was something we wanted to incorporate in our project, that level of the different scales, from micro to macro. So that was why it was so important that you saw the organism as a whole, but also you saw it on a cellular level as well, because we were really blown away by that. Like that we could see the organism from these different kind of levels.
It's kind of interesting because we have a project that it hasn't been kind of formally announced yet, but we're going to be working with cell biologists and And we've been really kind of thinking about how many kind of striking kind of comparisons can be made between our understanding of what's happening at the level of the cell inside the body and ways we think about health, and also ways we think about health if we start to consider ourselves as players, as individual components of a much larger system. And I think it kind of opens up some really interesting questions. And one of the things that we found quite striking is that when you think about the gaze of the scientist, is that working in a cell biology context is really directed by an idea of disease, an idea of what happens when a system stops working. That's a very kind of human preoccupation with health, that we're very kind of, we don't like the idea of our bodies stopping working. So it's the first time that we kind of start to really be interested in what happens at the level of the cell, is really kind of prompted by the fact that our bodies stop working, and it almost entirely consumes all of that energy is looking at cellular biology from the perspective of disease. And that's kind of a really interesting thought when you think about that how much is that distorting what we see and the understanding that we gain from that. And at the same time, if we really kind of twist that around and try and think about all the kind of problems that we face with kind of complex systems, human behavior, human consumption, and how we all play a part in that. Why is it that we can have an area of research that's so kind of focused in a particular way of thinking about it, but that insight stays locked inside this very kind of very narrow kind of framework of thought? So that's, and yeah, I mean, especially
I think that's what's so exceptional about the Arizona Cancer and Evolution Center, because with the psychology department, they're looking at it as, cancer from the perspective of collective behavior as well. So like system in the world, but also on a cellular level as well.
So for example, like things like autoimmune disease, it is a really kind of good analogy where it's where the internal mechanisms in our bodies suddenly become quite self-destructive. And that seems very, profound when we think about so much of our human behavior. The mechanisms of human behavior are becoming quite self-destructive, and the role of the immune system in recognizing that. How does an immune system tell when it should be kind of behaving in a particular way? How does it kind of recognize the difference between healthy kind of structures and healthy kind of behaviors, and when does that kind of intervene, and how do we draw the distinction between a system that is kind of is able to function to our benefit, and one that's actually kind of quite harmful?
Yeah. Oh man, it's always this way where the conversations just start getting really interesting, particularly since we're all dealing with COVID and against that backdrop. But it is our hour is over. So I just wanted to thank Paul and Vicky and Brittney and Angelo. Special thanks to ASU Art Museum and the National Cancer Institute, which is funding all of this. Mickey Garcia, Andrea Feller, Christopher Miller, Corey Rogers, all sorts of people from the ASU Art Museum who are doing amazing work. Special thanks to Fritz, who makes this all happen. And I also just wanted to say special thanks to Diem, Magda, Kamal, Mariella, Kuhn, Nick, Diane, Christiana, and all the other people that are here today. Lori, it's always good to see your face. And Jennifer, I'm really curious about your work. So tune in next month. We're not really sure what we're doing, but I'm sure we'll do something interesting. But meanwhile, stay safe. And thank you again for tuning in. Appreciate it. Bye.
Jennifer, feel free to contact us if you want to. Yeah.
Ditto.